Pour,
Ouais. Attends, OK. Oui, je te mets en CC dans le cours. J'enverrai merci.
Bonjour à tous. Bienvenue à la conférence midi de ce 31 octobre. Donc aujourd'hui, on a le plaisir d'accueillir Dr Nárlon C, Boa Sorte Silva, qui est un nouveau professeur dans le département de santé, kinésiologie, physiologie appliquée à l'université Concordia. Donc Dr Silva a été recruté récemment et commence son poste commencé son poste cet automne comme professeur adjoint. Donc Dr Silva a fait son stage postdoctoral à l'université de Colombie-Britannique, dans le laboratoire de Teresa Liu-Ambrose, où il s'est intéressé à travailler beaucoup au rôle de l'activité physique, mais aussi du sommeil et d'autres comportements de santé dans la promotion de la cognition pour le vieillissement. Et son programme de recherche utilise plusieurs types de modalités, donc il s'intéresse évidemment au comportement, mais il a fait aussi des études en neuroimagerie. Dr Silva va déposer une demande pour avoir une chaire de recherche du Canada j'imagine, en vieillissement actif et exercice. Je ne sais pas quelle va être la titre exacte de sa chaire, mais donc c'est un poste qui venait avec une chaire du Canada. Confiant qu'il pourra l'obtenir. Donc, sa conférence aujourd'hui s'inscrit dans le cadre d'un possible ou potentiel recrutement comme chercheur au centre de recherche du CIHR. Et donc maintenant, sans plus tarder, je vais passer la parole à Dr. Silva. So I'm going to switch to English. I was just introducing yourself. Because I don't know how familiar you are with French. Are you learning or?
I'm learning.
Okay.
Yeah, and I could grasp about half of what you said.
Okay, so just introducing yourself, like where you come from and what you're doing. So I was saying that this talk is also part of the potential affiliation for you at the center, and so this is hopefully the talk will give people some opportunity to discuss with you about potential collaborations and potential interest in you joining the center. So Nárlon is going to present a talk titled Promoting Brain Health via Active Aging from Midlife to Older Adulthood. So Nárlon, thank you for coming, and we look forward to listening to you now.
Yes, thank you for the invitation from the directors. It's a pleasure to be here. I wonder if people on Zoom can hear me okay? Yes, thank you. I see somebody. Okay, thank you. The talk, as described, will cover some of my work on exercise and brain health, and then what I want to do moving forward, which is where it relates to the center here, and hopefully we can brainstorm some ideas together if you guys think that whatever I'm doing is useful or interesting. I'm happy to elaborate as well on other research venues that I'm not talking about here today, but that I'm also involved. One of them is working on long-term consequences of COVID on brain health, which something I'm absolutely very interested on. So Concordia is located on indigenous lands. I wanted to acknowledge that these lands, which Concordia occupied, and historically belong to other peoples, and it's our duty to always remind ourselves of that. Thank you for the introduction. Briefly, I did my postdoc at UBC. This is the building. I always use that because I think it symbolizes a lot of the work that I did, but also the nature of the work. This is the Center for Brain Health. That was a generous donation by a constructor named Javad Mobafagan. He was a Persian constructor, and he developed Parkinson's disease towards the end of his life and decided to donate some of his fortune towards research on brain health. So we were very fortunate to have this place, and I got to do 4 years of my postdoc there, which was an amazing experience, and it's a place worth visiting if you are ever in Vancouver. Originally, I'm from Brazil. You have visited. It's a beautiful building, and I got to sit just beside a window like this to see the mountains in the back. It's just gotta cherish those things. Yes, and I'm from Brazil originally, born and raised, moved to Canada for my grad school. In Canada, I went to Western for a PhD, started as a master's, became a PhD, and then again to UBC for a postdoc. So today, no, at the beginning of the month, it's been about 8 years since I've moved to Canada. It's a long time. That's great. And now, like, it was sad. I'm an incoming professor at Concordia at the rank of assistant professor. I'm a nominee to the Canada Research Share program. I'm applying in April. Hopefully it goes through so we can have funding and some cool work. Very fortunate, very lucky to have been selected for this. So the selecting committee was very generous as well. I'm very glad.
I'm really sorry.
I don't apologize. I thought I would cover a little bit of my doctoral research, then a little bit of my postdoc research, and then what I would like to do next. I worked in different projects in my PhD at Western. A lot of them relate to exercise interventions for brain health, but also mobility, also cardiovascular health. And today, I'm just covering the one study that was the main study for my PhD. And it was published in PLOS One back in 2018. This study was a randomized trial that looked at exercise intervention on cognitive outcomes. It was a 6-month program. It was randomized, and we combined exercise intervention with cognitive challenging task. It's called square stepping. Was developed in Japan, and it's been used across many different research institutes. Canada and Brazil and other countries as well. The premise of the program is to have participants memorize stepping patterns and then reproduce them. The memorization followed by the reproduction would tap on cognitive domains that are usually related to working memory and executive function. Those are cognitive domains that decline with aging. And by stimulating this type of activity, we would think that it would result in improvements to cognition. But also spatial memory is one of the key components of this. The training had 2 arms. One arm did just the exercise, as is described in that figure. The other arm did the exercise plus the mind motor training. The intervention was balanced, so both groups had the same amount of time in the intervention. And then we saw that this intervention had a potential for improving cognition. So at the end of the 6-month program, we saw that both groups improved on cognition, which is typically expected for cognitively normal older adults. And we saw a trend for between group differences. And after another 6 months of a follow-up, the group that actually did the square stepping showed now cognitive benefits. So this overall suggests that by supplementing exercise with this cognitively challenging task, would benefit cognition in this particular population. And the main limitation of this trial at the time was that we didn't see the expected effect at the 6-month mark. So probably because the intervention stimulus was not intense enough or frequent, but then you run into issues of here's 3 days a week of exercise for 1 hour. How feasible is it to do 4 days of exercise a week for 1 hour? Not as feasible, perhaps. So it's a good compromise. We still see long-term benefits, which is good. So for my postdoc research, I decided to focus on some specific clinical groups at UBC. And I usually give this talk to students, to research audience, to people like you guys, which is a more refined research audience. And I do like to do a quick exercise, and I hope you don't mind. I just want you guys to close your eyes. Even people on Zoom, I'm watching you as well. Close your eyes. And then I just want you to imagine what the human brain looks like. Just a picture of the anatomy of the human brain, just standing in space, so as you would depict it. And then you can open your eyes, just nod your head if you feel like you saw something similar to this image. It's pink. You see it usually from a sagittal view. A lot of people see it from axial view, but it's usually sagittal because the side is depicted. All right, so the exercise that I do is to show it in a different perspective. And this is to show the vasculature of the brain. This is actually human vasculature from artwork from this guy from Germany. developed a technique called plastination that allowed him to preserve fine tissue using this technique. A lot of controversies about the technique because you're using human samples for artwork, and people don't like that in many countries. But it's a very interesting way to look at this. In any case, this serves the purpose of showing that blood vessels are everywhere in the brain, and any conditions or diseases that influence the blood vessels, they influence the brain. Nowadays, there's something called vascular cognitive impairment, which is an entity of, is this my screen? That's much better. All right, so vascular cognitive impairment is a condition that you guys may be familiar with. It's very broadly defined. It is the second most common cause of dementia. It accounts for about 15 to 30% of dementia cases on its own. Recent estimates suggest that at least half of dementia cases have vascular components to it. And especially in conditions like Alzheimer's disease, where you don't have Alzheimer's disease on its own, vascular is the second main player. And it refers to any degree of cognitive decline that is associated with changes in the vasculature of the brain. Typically, you would want to associate, you have a cascade of events, so you have some sort of vascular indication of decline, and then you see cognitive changes in domains that would make sense for that change in the brain. One example is frontal lobe functions are typically affected by vasculature changes in the brain. So executive function is one of them. One of the hallmarks of the disease is white matter hyperintensities. So these are all MRIs, and they all have different sequencing to them. This one is a flare sequence highlighted in purple that shows white matter hyperintensities indicated there as the bright voxels within the white matter. These hyperintensities usually, are associated with vascular function or some sort of vascular pathology. Really hard to know exactly what that pathology is, mostly because it's hard to mimic this in animal models because mice have no white matter, basically, compared to humans, proportionally speaking. A lot of the studies that we see for white matter pathology comes from histology. So brain samples of individuals that have died like a long time ago. So the understanding of the mechanisms is really not clear, but we just know that white matter hyperintensities are strongly associated with cognitive decline and predict dementia conversion and so on. So it's an interesting outcome. So a while back, I worked with people from different countries through the Alzheimer's Association in the United States. We published a paper on novel ideas on vascular cognitive impairment and how novel research could take advantage of these ideas and perhaps develop research in that direction. And again, this is the second most common cause of dementia on its own. It's also contributes to Alzheimer's disease. It's relevant to understand vasculature, related changes in the brain. These are risk factors associated with diagnosis of vascular cognitive impairment and dementia. Many levels of this relate to lifestyle, such as poor eating, poor sleeping, smoking, and lack of physical activity, which presents an opportunity for interventions. So in my postdoc work, I was trying to understand different factors that influence vascular health and brain health, generally speaking, in people that present with vascular cognitive impairment. There are a few papers here. I will highlight one of them because that's the one that to me is interesting. There's a paper that I'm currently working on, and I presented this work at the Alzheimer's Association this year. But just as a highlight of some of the papers that I have worked on with people through the Alzheimer's Association in the United States, all tap into different components of vascular cognitive impairment research. I come from the lifestyle exercise component, so that's where I want to, that's where my contribution is. But here we have people that work with basic research, people that work with biomarkers, others that work with clinical components like Dr. Fanielahi. So these are different papers that we have worked on. One of them actually looks at the SPRINT-MIND trial, which is a blood pressure reduction trial in the United States that led to cognitive prevention of cognitive decline via hypertension control, which was very interesting. We also, in that trial, saw that white matter hyperintensities reduced in size after the trial, which is also a positive outcome from this. So all of these papers highlight this work that I'm interested on in my postdoc and that I have contributed. I think they're all open access as well, so anybody can access them. One question is, can exercise slow down BCI pathology progression? And here, I mean, white matter hyperintensities. So the work at UBC by Dr. Liu-Ambrose a while back showed that aerobic training can improve cognitive health in people with BCI. But we don't really know if resistance training, which is a type of exercise that involves lifting weights and has a different metabolic component to it, would also benefit cognition. So the aerobic training program was published in Neurology in 2017. It's been very well received. So Teresa, at the time, Dr. Liu-Ambrose designed a program for resistance training because there are limitations with aerobic. As you know, people above the age of 65 have a high risk of falling or enduring injuries related to exercising because of balance controls and things like that. It's not also as easy to run at that age as it would a few decades earlier. So resistance training is a bit safer that way because it doesn't require a lot of ambulatory movement, so walking, running, and it has benefits to bone health, which is a key concern in aging. Resistance training is associated with IGF-1 release, and animal work will suggest that this leads to cerebrovascular improvements in vascular density, improvements in cerebral blood flow, and also myelination. And then likely also would result in reduced white matter hyperintensities because of these vascular changes. And there was some exploratory work published a while back in JAGS looking at the sample of females that underwent a year of training, and the program showed that people that did resistance training had a slower increase in white matter hyperintensities volume. So that was a good outcome, and it also correlated with gait. So after 12 months of training, white matter hyperintensity is reduced and gait improved, which was interesting for the context of aging. Gait is an important outcome as well. The work that I'm presenting here is the results of this resistance training program that was designed now for a larger sample. And the idea is, can resistance training slow down white matter lesion progression? And this is a randomized trial. We had about 90 participants at baseline. We had some dropouts and some quality control procedures that eliminated some participants from the MRI analysis because the data weren't of high quality. And we ended with around 74 participants for analysis. These are people that trained on weightlifting twice a week for an hour for 12 months. And
that's a huge sample size for those interventions. I know in neuroscience, it's we always say it's low sample size, but in the world of kinesiology, that this is a huge sample size. Having this sample size over 12 months is huge.
Yeah.
So I'm a kinesiology guy. Mm-hmm. So I'm always remind people that making such intervention is huge and difficult because it's unrealistic what the CIHR is asking us with the sample size. It's too much money, so it's awesome what you did. It's huge what you did.
It's a very large sample size. A sample size of people with white matter lesions in their brains, which requires that everybody had to go MRI screening before they could be enrolled in the study. So it took 7 years.
Yeah, it's huge.
But we got there. And so we used a pipeline to segment white matter hyperintensities. This is developed by Dr. Roger Tam at UBC as well. Originally developed for multiple sclerosis lesion segmentation, and it involves a trained expert placing seeds on the MRI scan, T2 and PD, the two kinds that we use, and the seeds grow to capture the whole lesion. It's fairly accurate. And the person doing this is a trained neurologist who would understand exactly the nature. Just this on its own takes a ton of time. That's why, again, it takes a lot of time to do this type of work. But just briefly on the analysis here. So the research question is, can 12 months of exercise training slow down white matter lesion progression? And we use ANCOVAs because it's a 2-time point trial design, and then we would add some covariates. But we also were interested in males and female differences because of previous evidence and because it's only fair to always report those results. All of this is done in R as well. But this is a sample what they look like at baseline. They're about 75 years of age and about 65% females. Always is the case in the aging population. It happens to be more females volunteering for these studies. The MoCA score, you guys are probably very familiar with it, indicating indicating MCI. And we tie the MCI with the evidence of vascular problems. We conclude that likely vascular cognitive impairment in this population. The MMSE indicates no dementia on average. And this is Vancouver. I'm not sure about Montreal, but in Vancouver, the population of volunteers for studies like this are highly educated. So it's incredible that 60% of our sample has university degrees, at least a university degree. So many of them have masters and PhDs as well. So this is a very specific population. And they all had around 4 comorbidities, such as hypertension or diabetes on the average. A lot of them had arthritis as well. So this is what we saw looking at white matter hyperintensities. We segmented it in different lobes. Looking at the whole brain, we didn't see any between group differences in white matter hyperintensities. But when we segment by lobes, we see that in the temporal lobe, there is a greater reduction of white matter hyperintensity than the resistance training group, which is the blue line there. And this is white matter hyperintensities has a poor distribution, so all of this is log transformed. But the effect is there, at least in the temporal lobe. But that is, we can see trends in each of the other lobes showing that the resistance training does lead to reduction of white matter lesions. But then we see something very interesting. We saw that females had an effect more so than males. So the blue line here shows females doing resistance training, and there's a reduction in white matter lesions, while females in the control condition did not. In fact, it increased a little. So it seems like the program might be more effective for females. So resistance training in females seem to lead to greater benefits to white matter health. And then you can go days discussing why this is the case, why females benefit from exercise training more than males in this outcome.
Can you remind us of the age range?
74 years of age is the sample average,
minimum
50 to 89, 55 to 89. So again, I presented this at the Alzheimer's Association, and the audience had a lot of questions about this. And one of them, one of them said, must be something to do with muscle mass in females versus males, and how much the training would influence muscle hypertrophy in females that in theory at baseline would start out with lower muscle mass just because of body composition differences between the sexes. That's a good idea. We do have muscle mass measured. I just haven't looked at it yet. Because part of the physiology of this is that if you lead to muscle hypertrophy, there is more release of growth factors because of the recovery. So the stimulus could be more intense for females than males, even if you dose it appropriately. Again, it's almost like there's more room in females because of that muscle mass component. Could also be because the sample of females is a bit larger. So we have a 60 to 40% split there.
You could also have the fact that based on the age of your population, it's likely that the women were less physically active during their overall, during their life, because if I'm correct, it's likely less work with their age, and it's also that they are involved in work with the resistance aspect, less involved. It's meaning that they didn't have the stimuli all over their life, instead that the men could likely have it.
That is, that's one of the probable reasons as well. Depends on the sort of, again, and then you enter the conversation about gender and roles in society, gender roles, and what sort of work and roles that men and women play, and other genders as well, which is a good point. And this is Vancouver. It's the most physically active city in North America. Maybe in the world, I think Australia, some cities there are a bit more active. But again, it's a very unique population. That's the main limitation of this. This is in preparation to be submitted for Alzheimer's and dementia as well. Hopefully we can get it out of the door soon. So this declining white matter lesions was related to the short performance physical battery. You guys probably use that a lot. I don't know the name in French, to be honest, but it measures overall out of 12 points, performance on a few physical tests. And the higher the points, the better the score. So reduction in white matter lesions in the temporal lobe were associated with an increase in the short performance physical battery. And that is a good outcome for us. And this is adjusted for potential co-founders like baseline performance and intracranial volume and the sex differences. So we do see that the program had an impact on the brain that might have led to actually some mechanistic relationship with function, which is good. We didn't see cognition though. That's the only thing. We measured cognition with ADAS-Cog and we didn't see a relationship. So one interesting question here is, you see white matter changes, at least in some lobes and mostly in females, but white matter lesions on the T1 or T2 image, they don't tell you about the pathology. It just says this voxel is not normal compared to the surroundings. And you can talk about what are the neurobiological correlates and can we measure it. So a lot of my work in my postdoc was trying to understand myelin as one of those components. So when you have a white matter lesion, a lot of it relates to myelin loss. At least that's what we see at post-mortem analysis of tissue and also in animal models. Myelin is this fatty layer that ensheaths the axons, and it's essential for brain health, for normal functioning. Multiple sclerosis is a condition that leads to loss of myelin associated with cognitive and motor function decline. So myelin is really interesting. It's hard to measure. It's very difficult to measure, but it's very relevant. It's very human as well. We have more of it than any other species, proportionally speaking. And it's one of the last additions to the human brain development through evolution. For those of us who believe in evolution, ends up being important. So you never know in which room you are. Scientific place, probably everybody believes in evolution.
Facts or belief.
There you go. So it's the last addition to the human brain, according to the people that study this. And it's very important in that sense. The myelin sheets are formed by this organ, this cell called oligodendrocytes, and you can have remyelination or you can have myelin loss depending on the health of the oligodendrocytes. And part of the idea behind vasocognitive impairment is that there is ischemia or hypoxia that prevents proper cell signaling, and then there is apoptosis of the oligodendrocytes, which then generates myelin loss. But there can be myelin regeneration because oligodendrocytes are cells that can still mature throughout adult life. So you have progenitor cells that can be stimulated for myelin to remyelinate. And if you put this in a conceptual model, is that you have cerebral small vessel disease, which is, again, a component of vascular cognitive impairment that leads to white matter pathology that is caused by this myelin loss. And then at the end, we see that manifest as cognitive impairment or mobility impairment. This is my understanding of it, and I'm no expert in any of this. I just use the techniques to answer exercise-related questions. So one paper that we published a couple of years ago was looking at whether myelin relates to physical activity levels in this sample. It's a sample of people with vascular cognitive impairment. We had a very picky reviewer that didn't want us to say vascular cognitive impairment, so we had to say people with cerebral small vessel disease and mild cognitive impairment in the title, because again, we didn't diagnose people. Always fun. And the idea is physical activity levels related to myelin. And we looked at a sample above 55 years of age. We had about 102 people. We had white matter lesions segmented, and these people were free of dementia. We use a pipeline that has 3 components. It's a T1 image. This is a T2 image that captures myelin signals. And then we can generate this myelin map. So the regions in yellow here is where you would see more myelin. So this is the occipital lobe, a lot of vision-related functions that need to be transferred really quickly from the eyes to the back of the brain. There's more myelin in those regions. And these regions here, for instance, are part of the cortical spinal tract that project signals from the motor cortex all the way down to the spinal tract. So there's a lot more myelin there as well. And hence why we see more myelin here. So what did we see in this particular study? It's a cross-sectional analysis. We see that people with higher levels of physical activity, this is measured using a questionnaire. The questionnaire captures not only leisure time activities like walking, things like that, but also tasks at home, housework, and gardening, things like that. So it's a very comprehensive tool to measure physical activity levels. And increasing the score in this scale of physical activity was associated with an increase in myelin levels in a few regions, which is a very good outcome. And again, this is controlled for sex, age, and other white matter lesions and other confounding factors. At least cross-sectionally, we see that if you're more physically active, you have more myelin in some regions of interest. But can exercise actually longitudinally cause that effect? And we used, well, this is based on animal research showing that if you put mice in different environments and look at the oligodendrocyte progenitor cells converging to full mature cells, what we see is that with stimulus, there is a greater myelination stimulus as well in the brain. So, mice that are exposed to exercise or social interaction do tend to have higher myelin rates in certain regions of the brain than rats that do not. So this is evidence that exercise as an environmental stimulus might be good for myelin. So we looked at the same population here, white matter lesions, were our key defining factor for cerebral small vessel disease and vascular cognitive impairment. The idea is, can exercise training for 12 months do this? It's the same trial, just a different type of data that I just showed you guys. I'm not going to go in depth about this, but this is basically what a year of my postdoc was, just trying to figure out how to do this. And that's why MRI work takes forever to get done. But again, we had to come up with our own pipelines because no one had before used this type of imaging sequence to look at exercise effects over time. Actually, no intervention, not even MS interventions did this yet. Basically, we measured myelin in these regions within the white matter. They are part of the Johns Hopkins Atlas that segments deep white matter structures. This is all done with a software called ANTS. I'm sure whoever works with neuroimaging might know what ANTS is. It's a very cool software. And the idea is to look at group differences over 12 months across these 15 regions and see if there are differences of the intervention. So we have the resistance training group against the active control group. And it's the same sample. So this table is the same as the one I showed before. This is what myelin looks like across the different ROIs. So you have, for instance, here the corpus callosum. This is the splenium of the corpus callosum. It's one of the regions with the highest levels of myelin. And here you have something like the cingulum where it's less myelin than the other regions. Just for fun, that's how it looks like. And then as it does in research, what happened was the exercise group, the resistance training group, did not show any significant changes in myelin, while the control group actually improved. And when we do the contrast between groups, we see that there's a significant differences, generally speaking, across all regions that we looked at, but also in two specific regions, here the corpus callosum and the fornix. So again, as a researcher, you look at this, I just want to pull your hairs out because I don't know how come this happens, given that the effect of the intervention of the lesions themselves was positive. One of the things here is that the control group actually did something. They did something very similar to yoga. It was a balance, relaxation, and mobility type of training that is a very good control for cognition, but may not be a good control for neuroimaging related outcomes. So these are people that, came in, spent an hour relaxing, socializing, stretching, making friends, getting out of the house. So there is an effect of the control. And the resistance training maybe was not the best, is not the best approach to look at myelin changes, even though it affects other white matter components, but not myelin per se. We didn't see any sex-related interactions here. But again, even looking at the data, it's a very good distribution of the data, not many outliers. Yes.
Just to be sure to follow, if you use the yoga, you still have movement and physical activity. So is it not more like it's more effective rather than it's effective? Because if you have a control group that is active, it changes. Is it not just more about testing about whether the effect is superior rather than the effect exists? It's just to be sure I follow.
Yeah, so in this case, some studies will have a third group that does nothing.
Okay.
And that third group expectedly wouldn't show any changes. From a trial standpoint, it's not a good idea to have a control group that does nothing because first it's unethical. You have to provide people some sort of positive intervention so it benefits their health. Even again, it's almost like we're providing them with a stimulus that would compensate them for their time, at least in the trial. So the people in the control group here, the active control, yes, they were very active. And again, this is Vancouver as well, so people are active on their own. The goal is when you have a control group that is active, you're trying to control for socialization, you're trying to control for changes in overall lifestyles like eating healthier. But the people in the control group did actually benefit from the intervention that they received. That benefit was not seen in white matter lesions, but it was seen in myelin health. And because it's a complex intervention that involves a lot of movements and learning and a lot of just more complex regions of the brain probably tapped by it. I just don't understand why the resistance training did not do the same thing.
Did you see anything behaviorally?
Yes, so the cognitive outcomes improved on the resistance training, improved on both, but there was an edge in the resistance training. That's the primary outcome that's almost done for submission now, but it's not something that I work directly with because I was focusing on the myelin and white matter lesion side. But yes, behaviorally, cognition was one of the outcomes that improved. Again, it's puzzling because you look at this in many different ways and it's hard to understand. Also, it's myelin. It's a novel technique to look at brain, and if it's gray matter, you understand that if gray matter grows, it's usually good. Gray matter shrinks, it's usually bad. But myelin, we don't understand it all that well. Is more myelin a sign of healthier tissue? Because there's also abnormal myelination in cases of schizophrenia, for instance, or seizures, where you have regions that are highly myelinated, but because of a pathological change, not because of physiological.
How about you correlate the NWF percentage to the cognitive outcome?
Yes, there was no correlation there, but there is with the physical one, the short performance physical battery. So again, the myelination in this case is positive. It's a good outcome. So it might be that the resistance training is not as important intervention for myelin, even though it helps with matter lesions. So just different effects that's happening there. This is humans. It's really hard to know exactly what's going on. Unfortunately, we can't sacrifice people. It's a joke. We don't do that. I don't want to do that. So actually, I got extra funding then to look at regions that are a little bit more complex, related to complex activities, because there I was looking at deep brain structures. Here, I'm trying to look at white matter regions that are related to specific cortical regions. It's called superficial white matter, because again, I was looking at deep white matter structures. It takes forever to figure out the pipeline, but here in yellow are regions that I'm interested on. It's just frontal lobe function. So the idea is the intervention did not have an effect as expected in the deep white matter structures. Maybe it is associated with an effect in myelin that serves complex cognitive functions, which we saw the effect of the intervention on. Still figuring out the code to run this. Mostly done, but have to finish. Thankfully, got funding from StrokeCog. This is the last part of my postdoc was working on this before I came to Concordia. This is a conference that I attended in February and part of StrokeCog. Anybody who's a postdoc, you guys have trainees that are working on this, probably know about the CTTP or the CAN train. So it's an opportunity for funding for people working in trials and research. So what's next? I will bore you guys now for another 10 minutes about what I would like to do next. Feel free to intervene. Dementia is still a problem. This is from 2017 from the Lancet Commission, showing that by 2050, we have still an increase in dementia, about 120 people around the world diagnosed with it, more so in low and middle income countries than in high income countries. And this is people living with dementia with a diagnosis, not people that actually have mild cognitive impairment. So this is somebody who's definitely living with dementia. If you put on top of that people that are living with mild cognitive impairment, these numbers are huge. Still a very big problem to be addressed, and there's still a lot of work to do in how to prevent. If you guys have followed the drug trials in the US using antibody therapies like aducanumab, donanemab, or the drugs that are being used, you see that they have very specific effects. It affects more males than females in some of the trials as well, and costs a fortune. So prevention is always a better solution. So there's still a lot of work to be done. So in 2017, this Lancet Commission highlighted risk factors that could be addressed in late life around age of 60 to 65 for preventing dementia. So they came up with these risk attribution factors. So 3% of the risk is associated with physical inactivity. So if you become physically active in older age, chances are that your dementia risk will be reduced at least by 3%. But exercise and physical activity also reduces depression rates and improves diabetes as well. So exercise or physical activity is a good holistic approach to all of this. So we all bought into this, and a lot of work has been done. But then they come up in 2024 with a different story. One of the main things that the Lancet Commission has done in the last 7 years is to basically say, at least for exercise, the longitudinal data, the UK Biobank data, all the observational data suggests that being physically active is great for your brain. Most randomized controlled trials say the opposite. So when you actually try to look for effects in randomized controlled trials, high quality trials show a positive effect of exercise. But on the average, the effect is very small, in the order of 0.23, which in the effect size world is very small. So, for instance, blood pressure reduction is in the order of 2, I think, the effect size is 1 to 2, and the exercise and cognition is 0.02. So it's really small.
But the effect of exercise on cognition, you're referring to the review in Nature Human Behavior recently posted?
That and a bunch of others, yeah.
Because this one, there is a serious letter of people from the center, Louis Berrer, Nicolas Berryman, and those guys who literally, I'm sorry to use this word, destroyed the review.
Yeah, and
it's a pure destruction of the review, and the people who publish this review, they have fun to just make systematic review and umbrella review without strong rationale and saying mental fatigue does not exist, TDCS doesn't work, exercise doesn't work. So I'm really cautious with those guys, because if you look at their CV, they only publish systematic review that show that doesn't work. And when they try to replicate experimentally, their stuff doesn't work methodologically. And also when they say it doesn't work, they have some correction drums saying that they say some mistake. So their stuff is really fun, make a lot of buzz on the social network. But as soon as there are serious researchers writing later, their stuff is destroyed systematically, and the answer is just, we did stats. But they don't have rationale behind.
Yeah, so that's a good point.
Sorry about that, but I think it's really important because I'm working on mental fatigue and their stuff is crap.
No, don't worry. So you guys are familiar with this paper that he's mentioning?
So we have we published a paper in Nature recently.
Yeah, so there's a paper in Nature Human Behavior published last year in April, I think, by a Spanish group that basically did a umbrella review of all the published trials on exercise and cognition across the lifespan from infancy to older adulthood. Well, they stop at 60, I think. And they look at healthy individuals only. So through their methods, they conclude that the effect of exercise on cognition is very small, 0.23. once you account for publication bias, the effect is negligible. That's how they word it. And this was published in a prestigious journal, so it made a huge impact. But no one who's ever done an exercise trial buys that story, and including myself.
I know. It's not against you.
Oh, yeah. So the way that they discuss the findings in the discussion is saying that because of publication bias, the exercise research on brain health is inherently flawed. So it's flawed, basically, and that's what people took to heart. And I agree, just saying that you guys do not do good research, and we must march forward with the ethics and with the unbiased way of doing research. However, the group that did this review, they've never done a trial. So nobody knows them in that sense, and which is a healthy thing in my mind, to go into a different field and do a very sophisticated review. But the way they approached the conclusions of their review was the wrong stuff.
But not only the conclusion, they did not include dissociation between kind of exercise.
Yeah.
They cherry pick the result they use in their study by considering that this is not a good test, this is a good test for no reason. It's all addressed, and they do the same thing in all field. And the sentence you use about this is flawed by publication bias, you copy paste this sentence, you place it's in all their stuff, and then you Control F, replace physical exercise, mental fatigue, TDCS.
Yeah.
So it's when you let's close this. The response led by Olivier Dupuis, and there's Nicolas Berryman, Hubert, and huge guys on that. It's
I know.
Ultra clear.
Yeah.
And their answer, just by the way, just about responding about statistic, but statistic is just a tool that you articulate based on a rationale, and they don't have rationale to do their stats in the way they do it.
That's right.
That's my issue.
Yeah, I agree. And I think this was a result of the pandemic. So once things shut down, a lot of people decided to do meta-analysis. And I think that group started doing that. Actually, I read the paper. I read the review report. I read all the comments from the reviewers because Nature published it. It's available. Because the first conclusion of the paper says that maybe the WHO should reconsider the recommendations for exercise and brain health in aging. And then the first thing all reviewers say is like, who are you to make recommendations to the WHO? Because that's what their conclusion was. But I think it's a healthy thing to, like, it's published, it's there, take the criticism where it serves you and then move forward. And that's sort of what I'm trying to do as well. Because regardless of whether the methods they used were good or flawed, nobody buys their conclusions either. Like, I think it's pretty bad. And I actually corresponded with, so there were, after this publication, there were letters that were written to criticize the paper by Louis and Sebastian Ludiga as well, was one of, and so I corresponded with Ludiga, like we sent a few emails about this. And what happened also, the editor of the publication was not actually, the editor was very biased. So the editor did not allow the researchers to actually reanalyze the data or republish the analyzed data. So they asked, can we reanalyze this and publish this? Like, no, only the authors can actually analyze and publish the analyzed data.
But that's an issue because when they did that with mental fatigue, some people reanalyzed and there was a correction drum and everything was wrong in it.
Yeah.
So that's it's anyway.
It goes against.
I have email from those guys like asking me effect size, and because they can't calculate it on a t-test, so it's like
back. It's really interesting what happens.
I have one question that it's not linked to this paper. You said that there was more than one review that goes towards at least a lower effect size than we would expect.
Yeah.
What are the others?
So there's one by the Cochrane. It's a Cochrane review, but a Cochrane group that yearly not yearly, but every 2-3 years they update. This one is looking at trials that measured cardiovascular function, like cardiorespiratory fitness and exercise and cognition. So the idea is, does exercise improve cognition in trials in which it improves cardiorespiratory fitness? So, and again, if you're biasing your sample because you're looking at data for trials that have both VO2 max, cardiorespiratory fitness, and cognition, and it concludes that there's no effect of aerobic training. So there's another review. It's more of a standing guideline from the European group that concludes that exercise is beneficial for overall health, but they cannot, put evidence of class 1 on its effectiveness for brain health. And that's what that review also mentions towards the end. But I guess my point is that the evidence is, and anyone who looks at animal research will be convinced that exercise benefits brain health. The human data is complex and requires larger trial. It's just a matter of time. I'll wrap up in 5 minutes so you guys can go back to your day. I just want to highlight a couple of things. So this is the review that I led with Professor Liu-Ambrose. We published in Trends Neurosciences. We made the cover, which was really cool. And here we talk about the evidence, and then we critically appraise what the evidence is like for brain health and exercise training. And we use the Syria review in Nature of Human Behavior as a point to criticize their work as well, but also to say, there are some components of this that would be healthy to think about, and how can we move forward. And then we came up with this figure that sort of shows what we know and what we don't know. I'm not going to go through the bullet points, but basically the evidence is small because the trials are complex. And you have complexity in the people that you recruit and how you deliver the intervention and where it's delivered. And if the effect size overall is 0.2, you need a sample of about 600 in each arm to see an effect. Yeah, so that's a criticism.
Give us 3 million.
But there's also novel ideas that you could explore. So you could actually better select your individuals. You could look at different people like in midlife instead of late life. Can also look at cross-sectional data and observational data and try to infer causation, which is something I'm interested in as well. So this is one of the gaps that we see right now, is that the evidence for exercise and brain health in midlife is scarce. There isn't much there. There's also not a lot of understanding about some mechanisms. So we know that BDNF, which is one of the main components of this, is well studied, but BDNF plays a ton of roles. Not only it releases muscle, it's released in the brain. It could generate growth in tumors, but also in healthy tissue. So it's complex. There are other factors like myokines, which is interesting. Most of the studies are done in North America or in Eastern Europe, Western Europe. So there's lack of diversity in the evidence that we see. We don't know if people with chronic conditions benefit the same, diabetes, hypertension, which is the majority of people. And so my idea is to sort of take a different approach now moving forward to try and look at aging as a different perspective. So when we talk about diversity and brain health and interventions for brain health, the brain is the most plastic organ and it responds to the environment. These are all pictures of what aging looks like across different cultures. And obviously this, basically you're looking at different brains. That's all we're looking at here. And life exposure, life changes influence how people age and how their brains age. And so the problem that I see in the literature is when you look at life in the older stage, most of the research talk about aging as if it is a property of individuals above the age of 60. Aging is a process, it's a biological process. So in my mind, looking at midlife is where it makes most sense. And so what I want to understand is, what does midlife can attribute, or what can attribute to healthy aging? Because basically that's what we're looking at, right? So there are different risk factors for brain health, and aging is the largest and most important risk factor for brain health decline. It's the strongest risk factor for brain health decline. So can you improve aging? Can you age healthier? But again, I think it's not, we need to shift the conception of aging for a stage and understand it as a process. So aging occurs, you would argue that people start to age at age 30, not at age 60.
Some don't even argue that it starts from birth.
And some people argue that aging starts at birth.
Yeah.
So my idea is, what about trying to study aging in midlife and see if interventions are effective there? A while back, the Lancet Commission said that in midlife, hypertension and obesity were important risk factors for dementia. So if you address that in midlife, you're golden. Their updated number shows midlife. Now you have cholesterol, you have physical inactivity, you have diabetes, you have hypertension, you have obesity, and you have all the others. So the literature is evolving and showing that midlife is perhaps a more critical point for brain health prevention. because again, the evidence that comes from observational studies in midlife seems to be more, consistently clear in that sense. And this is what before old late life was considered, like all those factors, smoking, depression, and diabetes were all in late life. And now they shifted back to midlife. So what I'm saying is that understanding aging as a process and looking at midlife as a starting point could be a healthy approach for studying brain health. And so late life right now for the Lancet Commission looks like this. So these are the factors that if you wanted to promote brain health, these factors being addressed in late life show the most promising results. Obviously, once somebody reaches 60, you're not going to tell them not to exercise, but maybe starting exercise at 60 may not lead to the strong effects that we would want. That's basically what this says. And the Lancet Commission is a very respected panel of researchers that thought critically about this. So it's really cool what they do. And I was at them to move to rural populations. Yeah, that's
it's really interesting, but I love it. It's just that I get lost in the reasoning, not yours, but their reasoning, because physical activity has a good effect on fighting air pollution effect. Social isolation, you are socially isolated if you cannot move. So physical activity allow you to have the social isolation. All the aspect below, if you come back in the middle life stuff, it's written there is physical inactivity, true, but physical inactivity lowers LDL, fight depression, it is tons of evidence. So this is why the whole rationale on this is hard for me to follow.
Yeah, I think their point here is that these are attributable risk factors, I think they're called, and they are independent, so they account for the other ones. But if you were to design interventions, starting with something that's more realistic, probably likely more efficient. So, instead of trying to reduce weight by administering a diet, maybe combining diet and exercise, you actually tap on other things as well, because dieting doesn't relate to depression risk, but exercise does.
And that goes with early age as well. We know that not in the Nature Human Behavioral Study, of course, but it's known that physical activity has a good effect on the development of cognition in young.
Yes.
So it means you increase the reserve since the young age, so you have a positive effect later.
Yeah, you predict academic achievement.
Yeah. So the problem is it's because I think the problem is physical activity is always seen as a medication, even so it's not that you give to people when they're not good, doing well. The problem is like we are tuned to be physically active. So if we are not physically active, we have all of that. So it's more an intervention. It's more like a daily behavior we should have.
Yeah, you're right. And so part of what I'm trying to understand is, if you look at midlife and you say somebody with hypertension has higher risk of dementia, great. But the conversion rate is not clear. 100% of people with hypertension do not get dementia. So a percentage of people with hypertension get dementia. Or a percentage of people with diabetes would get dementia. So how do you tell who is at highest risk and who is not? Even if you got a group of individuals that have several risk factors, and then everybody has diabetes, hypertension, high cholesterol. Of those, not every single one of them will go on to develop dementia. So there is variability in that risk. And so that's why it's not a one-to-one relationship. How can you actually identify those people? And, and of those that you identify, who would respond to an intervention? Because exercise is great, but there's variability in the biological response to it, that some people benefit greatly, some do not. Some people are great marathon runners. Some people can't run for it. Does that make sense? So there's variability in how physiologically we respond to training, because there is, by natural selection, for those of us that believe in it, there's variability in how we evolve, and that variability actually plays a role in how we respond to environmental stimuli. Going back to midlife, why is midlife important? I think looking at midlife makes more sense for brain health. And so how can we identify the high-risk people? In my mind, we can use a lot of tools. Nowadays, you can use machine learning, which is a component of AI. And this is really cool because you can use machine learning for disease staging. So if you have a bunch of people with a condition, you can actually use these tools to better characterize in which stage they are. You can also detect a risk and stratify people according to that risk. Again, everybody with hypertension is at risk, within those within hypertension, who is at the highest risk? Because everybody's already got hypertension. What else can you extract from the information you have from those people? And you cannot do 20 or 20,000 regressions to find this effect. You can't do that. You cannot do a matrix by matrix regression where you have like 400 variables in one end, 400 variables in the other end, and find every single interaction to see who's at highest risk. Machine learning is that level of regression being applied.
Now, what if there were two people who just left? Does a bit of that.
There you go. It would have been cool to see them here. But again, I'm not an expert in machine learning. Not at all. I'm not an expert in MRI either, but there you go. I can use it. I'm an enthusiast, yes. So I'm an enthusiast. I like it, and I think it's cool. Disease staging with machine learning is one of the cool things that I've seen. This is from 2018, but there are developments now. This one tool gets MRI scans, it gets cortical thickness, the cortical volume, and then it can look at a sample of healthy controls and say, this person has atrophy in this region, and they belong to a group of people that have similar atrophy patterns.
Have you used Sustain?
I have not used Sustain. Again, I've read.
No, we need help, that's why.
I know how to run it.
No, we also know. Yeah, we can discuss that.
Yeah, because you got Alexander Young there that I think they're very responsive if you guys reach out to them. I always reach out to these people. Hey, I cannot use your software. Help me. But Sustain is one of them. So it basically stages people along a continuum using cross-sectional data. That's the coolest thing of it all. It doesn't need time-related data. It's cross-sectional. That's what it does. And if you really like this, you guys might have seen this paper by Alexandra, but also other people like Nick Fox and etc. That here discuss how can you model disease in aging, brain-related disease, of course, using different techniques, and machine learning is one of them. So, but this is what, in essence, you can. Let me go back to that one if you need a photo. There you go. Really, really cool paper. It takes a few tries to understand it, but it does really work. So what Sustain does, and many of the models that are developed, they have snapshots in time of people, and then they can project them over time, even though they do not have the overtime. But the idea is, if you have a 60-year-old and you have a 90-year-old in the same sample, those two people could be following the progression of the same disease, so they represent the different time points that you need. So if you have somebody at 60, 65, 70, 80, you can project, you can model better, saying their progression, and then you can look at different biomarkers. So each color here is a biomarker that you can look at. And then each square, each symbol here is an individual along that biomarker curve. So the idea is very simple, hard to execute, hard to develop, but it's simple in essence. Would I be able to develop this? No, but can I actually operate? It's like looking at Michelangelo's work and be like, this is really cool. Would I be able to do that? No. But there are some applications that are a bit more tangible as well. So this one is developed at UPenn, and this guy is just amazing. He developed this tool that, in this particular case, it looks at white matter hyperintensities in people with hypertension. Everybody here has got hypertension, all the same. But then when you look at their brains using this algorithm, you can find that some of those people have higher, clearer patterns of atrophy than others. Everybody's got hypertension all the same, but you cannot tell, just looking at the hypertension numbers or any other characteristic, which of those have higher risk. That's basically what this shows, and it's really cool. And this is available on GitHub. It's easy to run, I think. And so this guy, he actually improved on the algorithm and published in Nature Medicine a couple of months ago an updated version of this algorithm. We have, again, gray matter volume data that you can process through his surreal GAN model. And then it gives you patterns that, or it basically identifies people along an index and say, these people are within the R1 index, and because they are within R1, this is a sort of pattern of atrophy we see, and they're more likely to have dementia of this type, or they're more likely to have any sort of cognitive pattern, or decline of cognitive patterns. It's really cool, really, really cool. And this is available again on GitHub that can be accessed. And this is using data from the UK Biobank and other research centers. The lab where this comes from has an entire pipeline that can be applied. I'm not sure if you guys are familiar with the lab at UPenn is CIPCA, C-I-P-C-A. And they have basically all the different tools that you can apply to get to these results on your own data.
So here we have people who, they don't necessarily do this exactly, but we have people who can understand this completely.
Yeah, it's amazing.
That we're happy to portray.
Yeah, and that's what I want to do. Yeah, so another cool thing, if you don't like MRI, you can look at blood-based biomarkers. This is what puzzles me. So this is a second-year PhD student, granted at a very well-known institution working with a genius, Tony Weiss-Corray's genius level type of person. But this guy is second year. In my PhD, I did my best. But this kid's got 2 publications already using this machine learning algorithm. One in Nature Communications, the other one is coming up in Nature. And this is what this kid did. He looked at protein levels in the blood that were related to different organs. And he was able to derive these measures and identify, basically using the protein biomarkers, who would be at higher risk of developing dementia or developing cancer or developing multiple sclerosis using the UK Biobank data. Really, really cool. This is a preprint, but again, I think it's going to get published soon. This is the Nature Communications paper. So basically, again, the premise is you measure the plasma proteins, and then you can compare within a sample whose protein levels are more likely to be older or younger than their own biological age, and then chronological age rather. Then you can derive patterns from that and be like, anyone whose age is above a certain threshold, anyone who's older than their proteins indicate, means that they're healthier because biologically they're younger, and the opposite is true as well. Again, just passing over this because it's really cool stuff. And then you can. Which paper? This is this. So this is the preprint. Okay. Yeah, you guys already got that. Yeah. So my idea is, okay, what can this do? Can we design interventions based on this? Because that's the application that I think is useful from machine learning. Is that I do not need to know how to develop the algorithm. I do not need to know how to, collect MRI data. But can we get expertise that help designing interventions based on this? My idea is get the people that you see is the highest risk possible to identify. Among them, would they respond to any intervention? Because when you sample randomly from the population, you get variability that is inherent to that.
That has been the problem with some of the neurodegenerative problems, including Parkinson's.
Yes. So, and then if you can better characterize them, your power increases drastically, because then you have a group of people that would respond, and then you need 30 instead of 300 of a sample. If you can better characterize those people, these machines.
Then we could have any CIHR.
Yes, CIHR. So the idea is, can we do this? Is it feasible at all? I met with somebody from UPenn a couple of weeks ago discussing this, and they're like, yeah, I think it's doable.
So your idea is to do a combination of these things. So you'd first do the modeling and find out who's more likely to respond, and then actually run the trial.
Yeah, based on that, that's
and the multimodal trial.
And again, yes, I actually want to look at cross-sectional analysis of data and see what intervention would more likely to be beneficial. Again, I'm interested in exercise stuff, so we'll probably go that route. But I know for some people, improving sleep quality might be more beneficial than for other people, right?
And any exercise improves sleep?
I think so.
Yeah, perfect. Easy.
Yeah. So again, I do not have experience using these machine learning tools. I think it's just as a tool to an end. People make their lives out of doing machine learning research, and that's obviously very noble. But the point of what they're doing is, can we actually apply it? And then it was the same thing with MRI. People got Nobel Prize in physics for developing MRI technology, but this serves a point, right? Serves an end. That's what I want to do. Just be in touch with the right people. I do, however, have experience with developing trials in midlife. So this is a trial that I developed with Dr. Petrella at Western. The premise is, overweight men that do not exercise at all and have poor healthy habits. And those men are still within the working force. They are 35 to 60. Obviously, people above 60 still in the working force. This is a higher proportion. And they're all hockey fans. So the premise is, come talk about hockey, and then we can talk about, you know, lifestyle change and healthy eating. This is done across many provinces in Canada. Me, a Brazilian, understanding a lot about hockey. Can you imagine how that went? I do not understand. Got funding from CIHR, from PHAC to do this. And the primary outcome was weight loss. And we saw after 3 months of intervention once a week for 1 hour that there was weight loss in this group. And the weight loss is basically by cutting, you know, back beer, you know, eating more vegetables and stuff like that. And actually,
hockey night?
Yeah, actually, some of the sessions were at a hockey team arena. So unfortunately, Quebec was the one province that we couldn't get a team to participate, like Saskatchewan as well. But like, you know, in Ontario, you have teams like the London Knights, which is like a local minor hockey league team. So fans of the London Knights would go to the arena once a month, and then there they'll get in touch with some of the players, they'll get to the locker room, and then they'll do this type of intervention, which is, okay, let's talk about health now. And just, again, because it's a population that is very specific, it's men. Why men in the first place? Men are stupid, generally speaking. Men are highest risk for developing cardiovascular disease in midlife than women are. Later in life, that changes, but in midlife at least, and strokes, all of that, higher rates of obesity among men and women, so targeting men. And women volunteer more for lifestyle change programs, men do not, so targeting men in this case. Anyway, so the results the paper got accepted in clinical medicine, and it's going to get published soon. But again, we saw a change in VO2 max, a change in weight loss. Anyway, so it's doable. We can target that population. That's basically what I want to say. I'm boring you now, but I'm just going to cruise through this. What I want to do is the UK Biobank data first and apply one of these machine learning algorithms. I don't know if Surreal GAN or Sustain or whatever, to find people that have signatures of brain atrophy related to function to vascular changes. So again, I'll target people living with obesity, diabetes, or hypertension, and then try to identify among those people with highest risk based on higher or greater atrophy patterns, and then look what actually is different among those people, and then see if physical activity levels moderate any associations there. Basically, okay, so you're the highest risk group, you have greater levels of atrophy. Among you, are the physically active ones still benefiting from that physical activity levels? If so, by how much? And what are the patterns of physical activity? Because the UK Biobank has physical activity data from objectively measured sources, so you can actually map daily levels very easily. And then, again, just saying that UK Biobank has all of that. It can use Alliance of Canada to do this type of work.
If you can access it.
The UK Biobank? Yes, I'm actually going through that right now.
We've been going through the last 6 months, and I've already paid, but I still haven't signed all the DTA.
My God, yeah. So, and the idea is something similar, is to recruit a similar population here, and then if, let's say, physical activity on the order of twice a week is enough to see a difference, for sectionally, maybe twice a week physical activity intervention would be good. And then that could be promoting physical activity. It could be exercise specifically, and then collect data on that. And then, you know, there's people that, like Simon, for instance, that works on behavior change. That could be useful to have their expertise in how to design these interventions so they're actually successful, have a higher chance. I'm not a behavioral researcher, but knowing somebody who is really helps. Okay, so, and the cool thing about Montreal is this is one of the reasons why I actually wanted to come here to work. Look how diverse this city is. There are nearly 2,000 people that self-identify as Black in this city. And then you have people that are out, you have Black Americans. Obviously, this is the visible minorities, and everybody else is quote unquote white. So this is a place to do this type of work. Toronto is not like this as much. also the francophone.
This neighborhood in particular.
Yeah, so there is really a logical way of doing this. Again, maybe the machine learning thing doesn't work out. Maybe you need so much computing power that it's not just feasible. Like at UBC, I'm working with a cluster that does a lot of it, but it's still very limited.
We are equipped to calculate Quebec here. They work with them, so they are.
Yeah,
we have enough capability here to run that.
I don't think that will be the biggest
Not
that's good. Yeah, because that was the big problem, and so recently where GPUs became a thing, right?
Calculate Quebec is
Yes, there could be some methodological issues, which Alexa maybe.
Oh yes, no her work. So anyway, so here's my disclaimer about this.
Well, one challenge here is recruitment, because the diversity in the population doesn't mean that we can reach out to get them. So I think your initiative with the hockey fans, I think it's really original to try to get where people are, where their interests in.
Yeah, and that's why, again, nobody should work in silos, right? So you need people that can do that stuff.
Link to that, we have people who work with the community, with associations. Actually, we have what's called a living lab here in.
That's right. Yes, I've heard of that.
Yeah, so that would help with those issues. Doesn't solve them, but it helps.
Yeah, and I think as much effort needs to go into addressing that particular problem as it is in running everything else, because at the end of the day, it serves nobody to have a trial published on a population that's not representative of the population, on a sample that's not representative of the population because of those biases. Again, it's a very challenging issue, and that's why you need experts in that. Again, my disclaimer is the only thing I do well is design exercise interventions. That I can do. No.
Montreal is a good place for deep learning, so it is a very nice. And your step here.
I apologize, I missed the beginning. I really apologize. So this is my question, maybe completely naive. So what do you mean by you have expertise to design? What's your actual background? I'm sorry, it's a naive.
It's all good. It's exercise research. Basically, I've designed 4 different RCTs and conducted.
So you created the programs, you exercise. Okay, that's super cool.
I mean, by training, I've been a personal trainer, I've been a coach, I've been trained myself when I was at university. I just love the research in aging and brain health, and again, reading and again, collaborating with people that understand those actually makes it more tangible. Again, if you tell me how you can go from K-space and MRI to the actual image, I will not be able to explain that to you.
I apologize for the naive question. It's interesting because
again, it's knowing that
for this expertise.
Yeah.
I know.
We are discussing.
I know.
Yeah.
But I didn't realize that was particularly your expertise, because that's exactly what we're currently looking for.
There you go. And my main disclaimer is none of this may work, but that's what research is for.
Yeah,
that's
cool.
That's life. It's data.
Somebody who really knows, because I talk to people that developed real gantries, and they're like, yeah, it's totally doable. I'm like, okay, in your mind, it's totally doable, but how can we actually make it happen? Or again, my point is maybe in 5 years, we can be like, well, you're just dreaming too big, or it just makes no sense. But I have all the passion to do it because I think that's how it should be done.
No, it fits, all you've presented fits extremely well with our new priorities in the center. It's really in the center of many of them. So,
and no result is a result. The absence of a result is a result as well. So
well, obviously, yes.
Even so, it doesn't work. It's result.
Yeah. So
it's not Nature. It's another journal, but it's results. It's good.
Yeah, I think I'm sorry, I've taken almost an hour and a half of your time, but again, I'll go
want to carry on with the lunchbox, maybe?
Yes, let's have food. I'm starving. Well, thank you for staying until the end. Sorry if that was too boring. I'm not sure if people on Zoom had questions, and I'm sorry if I missed those questions. I think I'm also benefiting.
And also to the great presentation.
Yeah, that's super cool.
Thank you so much. Oh yeah, I'll stop the recording here so we don't go